seminar. I'm a PhD student, as I was mentioning. I'm a PhD student at the National Autonomous University of Mexico, and I will be happy of moderating today's seminar. I am also part of Open Book Science, which is a nonprofit organization that is main goal is to promote science and scientific discussion free and open to anyone. And I'm very happy that we could have the opportunity to do this joint seminar today, as well with the junior iOS community. The junior iOS community is also is focused on promoting immunology discussion. And I was told today by Marion that we have upcoming events that if you are part of the Mexican Society of Immunology, you will be also part of the iOS junior community. So we're very happy to have this community growing, and hopefully we can have more of these events today. So today I'm very happy to present Dr. Geoffrey Lamet. Geoffrey obtained his PhD from the Pasteur Institute in Lille, France. He completed his postdoctoral training at the University of Texas MD Anderson Cancer Center before joining the faculty of Michigan State University. He's currently an associate professor in the Department of Physiology and a member of the neuroscience program at Michigan State University, where he leads the neuroimmunology of pain lab. His research focuses on how interactions between the nervous and immune system regulate the transition from acute to chronic pain. Dr. Lament's research is supported by multiple NIH and Department of Defense grants, and he's a Rita Allen Foundation Scholar. And today he's going to present one of his latest work on pain. And I'm happy to say that Maria Fernanda Aguilar, at the end of the session, will be also moderating the Q&A. She's a brilliant PhD student, and she's very happy as well to be with us. And Dr. Lamet, thank you so much for accepting the invitation, and the room is yours.
Thank you so much. Thank you so much for the kind introduction. I'm very glad to be here at the opportunity to present in front of the three communities: the Open Book Science, the Mexican Society for Immunology, and the International Society of Young Immunologists. Okay. Yep. So you can see my screen.
Yes.
Okay, perfect. So thank you everyone for being here. I will quickly start by presenting my lab. I will try a couple of words in Spanish, and then we'll dive into the paper. So buenas tardes a todas y todos. Estoy muy contento de estar aquí. So muchas gracias for this opportunity. I will go back in English to stop terrorizing Mexican fellow. So my lab studies the interaction between immune cell and sensory neuron, and we're doing that in mouse model to see how that interaction can shape the trajectory of pain. So to give you a bit of background, because I guess that most of you might not be super familiar with pain and also with neuroimmunology. So sensory neurons are neurons that, like you would see in blue, that basically innervate every part of your body. To make it simple, if one part of your body can feel pain, that means it's innervated by this sensory neuron. So we're really interested by the communication between immune cell and sensory neuron, especially at the peripheral terminal of this neuron. So where the neuron and in the end organ, because this neuron will rely, for example, here the skin to the spinal cord. But for my lab, we're really focusing on that part at the peripheral terminal of the neuron and how it talks with the immune cell. The communication with immune cell is really bidirectional. I show you here some examples that from previous literature. That's a review we wrote 2 years ago. The sensory neurons are actually able to sense a wide range of immunological stimulation. They have receptor for cytokine, receptor for chemokine. They also have some TLR receptor. They have FC receptor as well. And activation of all of this receptor by the specific ligand will change the activity of different ion channels. And that will modulate the excitability of the neuron and its ability to generate action potential, which is the way for neuron to communicate to the next neuron and to convey information. So some immune mediator will increase the ability of neuron to generate action potential, and some will actually reduce that ability. And so it's a really plastic way for which on how the immune system can influence neuronal physiology. And as I say, the communication go also in the other way. Sensory neuron can also release neuropeptide, for which we found a receptor on a lot of immune cell. One of the well-known is substance P, which one of the natural agonists for the receptor MRG PRB2, so it's a difficult name, that is on mast cell, and so substance P can induce mast cell degranulation. But the sensory neuron can also release chemokine like CCL2 to attract immune cell, CSF1 to activate macrophage, and also TAFA4, which will also modulate immune cell response. So both the immune cell can modulate neuronal physiology, and sensory neuron can modulate the immune response. So in my lab, we really focus in two places to look at this interaction. The first one is in the skin, because the skin, as you may know, is very densely populated by immune cells. And it's also innervated by sensory neuron. And that's when we look at somatic pain, and I will talk a lot about that today. And we have another axis in the lab looking at migraine or headache. And because the sensory neuron in the head will innervate the meninges, which you can see here. And when you see the neuron in green and immune cell are in red, and on the other side, that's the lymphatic vessel, that's also important for that communication. But so you also communication of between sensory neuron and immune cell in the meninges that we thought play an important role on migraine. Okay, so now I will start really going into the topic of the talk today. I will talk about the immune basis of sex difference in pain, and mostly about that work that we recently published in Science Immunology. So chronic pain has actually in the US a bigger cost to society than diabetes, heart disease, and cancer together. But despite this super high prevalence, less than half of the patients actually receive adequate pain relief. And currently in the US, the opioid crisis that really come for our inability to manage chronic pain is now killing in the US more people than HIV in the 80s. So there's a really need to improve our way to manage chronic pain. And we think that for that, we need a better understanding of what actually regulates that transition from acute to chronic pain. So because I think most of you are immunologists, I wanted to give you a brief introduction on the neuroanatomy of pain. So we have sensory neuron that I may also call DRG neuron because they have the cell body in the DRG, the dorsal root ganglion. On the peripheral terminal, those neurons will detect painful stimulation and will project to the spinal cord when a neuronal circuitry will go to the brain to bring the information about the experience of pain in the brain. But for today's talk, we really focus on that sensory neuron that is here. And so that's what it looks like in mice. We have the dorsal root ganglion that are here very close to the spinal cord and the sensory neuron. Here I will specially focus on the one that innervates the hindpaw. So we have known for some time that those DRG neuron or sensory neuron can be excited by inflammatory molecule such as pro-inflammatory cytokine, ROS, or prostaglandin. And actually, that's one of the reasons you would take something like ibuprofen when you get hurt. Ibuprofen will block the synthesis of prostaglandin, and so reduction of prostaglandin will prevent prostaglandin to activate those sensory neuron and will prevent pain signaling. But we knew that for a while, but a few years ago, we had no idea of what was the impact of an anti-inflammatory molecule on the excitability of sensory neuron. So I wanted to know if interleukin-10, because it's a major anti-inflammatory player, would have a role on sensory neuron. And so to do that, we treat mice with cisplatin or PBS as control. So cisplatin is a chemotherapeutic agent that is well known to induce neuropathic pain in patients. And neuropathic pain is really associated with hyperactivity of the sensory neuron. So 10 days after cisplatin or PBS injection into the mice, we harvest the sensory neuron and we measure the electrical activity with patch clamp recording. That was done by Dr. Bavenkov in Houston. And we add interleukin-10 in the culture medium. So sensory neuron isolated from PBS-treated mice, they show no activity. That's expected because in absence of painful stimulation, those neurons remain silent. Neuron from cisplatin-treated mice show spontaneous activity. And so that means that they will induce a pain signaling even in absence of painful stimulation. That has been also observed in human patients with neuropathic pain. But when if we treat the neuron with interleukin-10, we drastically reduce the spontaneous firing of those neurons. So that was the first demonstration that an anti-inflammatory cytokine can actually reverse neuronal hyperactivity in sensory neuron that activities that are associated with chronic pain. I'll skip that one. So a fact that is very important to know about chronic pain is that women are much more likely to develop chronic pain than men. And that's true regardless of age. That's so true regardless of culture, and so which would associate with social construct and race. And it's also true regardless of the painful condition. And here on this graph, what you see, if the bar go up in the red part, it's more prevalent in women. If it's go in the blue, it's more prevalent in men. And you can see that in all of those different type of painful condition, it's always much more prevalent in women. So therefore, we need to reduce that inequality in pain and pain management. And so for that, it's important to understand the mechanism that is underlying that sexual dimorphism. Interestingly, we know that the immune system is also sexually dimorphic. And you know that, and it's basically stronger in women and female. With women are much more likely to develop autoimmune disease or allergy, while men are more likely to get infection or non-hormonal cancer. And so we wanted to test the hypothesis that the sex difference in pain may actually result from sex difference in the immune system. So to do that, we use a classical inflammatory pain model. When we inject complete thread adjuvant into one pore, and we inject saline in the contralateral pore. So saline is our control, does not do much, and CFA will induce a strong inflammation, and the pore will have swelling, increase its thickness. That's what you can see here. When we measure the increase of thickness with the pore edema, with no difference between male and female. So then we look at pain. So to look at pain, we're measuring mechanical sensitivity by measuring the force that is required from calibrated filament for the mice to withdraw the pore. So to make that simple, when they're up, it's less pain, and when the bottom of the graph, it's more pain. So at baseline, male and female mice have about the same, have the similar level of pain. When we inject them with CFA, they both develop a drastic, very severe pain hypersensitivity. But as you can see over time, male mice will resolve pain faster than female. So because of our previous work, when we show that IL-10 can regulate the activity of sensory neuron, we wanted to see if IL-10 was different between male and female. And so that most of the work that I will present is actually being produced by a fantastic graduate student, Jiwon Sim, that she just graduated and left my lab to start a postdoc. So because interleukin-10 is very difficult to stain with antibody, we took advantage of an IL-10 GFP reporter mice. And here, so we measure by flow cytometry of immune cell in the skin of the hindpaw. As you can see, there's very few immune cells that express GFP in paws that have been injected with saline. But in inflammatory conditions, there are huge increase of immune cell and also huge increase of GFP positive cell, which are the IL-10 producing cell. So while CFA increase IL-10 producing cell, that increase was substantially stronger in male compared to female mice. And when we did qPCR on skin samples, we observe exactly the same trend with CFA induce a strong increase in IL-10 expression, but that increase is stronger in males than in female. So we are interested by neuroimmune interaction. So if we have immune cell producing IL-10 in the skin, we need to have sensory neuron expressing IL-10 receptor in the skin as well. So that's one of the things that we confirm. We can see here in the skin that we do have sensory neuron that express IL-10 receptor. We also look at that in the DRG, so that's where we have the cell body of those neurons that innervate the skin. You can see that nu-N, it's a neuronal marker. IL-10 receptor is in green. So cells that express both red and green, so that they turn like orange-ish here, are cells that are neuron that express IL-10 receptor. We've done more work that I will not go in detail into it to characterize exactly which subset of neuron express IL-10 receptor. And in mice, we found that it was that neurons that express IL-10 receptor are non-peptidergic P2X3 positive neuron. So that we wanted to validate that if our staining was specific. And so to do that, we generate transgenic mice using Advil-Cree, which Advil-Cree, it's a Cree driver that is specific for sensory neuron, and we cross it with IL-10 receptor flocks. So in those mice, we remove IL-10 receptor specifically into the sensory neuron. We could confirm that. You can see here in the skin, they have nerves that express IL-10 receptor, and that staining disappear in our knockout mice. And we look in the DRG. We see that those neurons that express IL-10 receptor also are gone in the wild type. I mean, the neurons are not gone, but there's no neuron that express IL-10 receptor anymore. We still do see some green positive cell. Those are either immune cell or glial cell. So which actually shows that our depletion was specific to the sensory neuron and did not affect other type of cell. So here we did show that there are sensory neurons innervating the skin that do express IL-10 receptor, and we can specifically remove IL-10 receptor from this neuron. So going back to our model of communication between immune cell releasing IL-10 to sensory neuron that express IL-10 receptor, when we use this, so if we treat mice with CFA, as we've seen before, it does induce pain, a strong pain hypersensitivity that resolves faster in male than in female. But in mice, when we remove IL-10 receptor, it completely impairs the resolution of pain. So those mice stay in pain for very long time, and that was true in both sexes. So we remove the receptor. Then we wanted to see what happened if we block the ligand. So here we actually inject a neutralizing anti-IL-10 antibody, and blocking IL-10 block also the resolution of pain. Then we try the reverse experiment. Here we inject recombinant IL-10 into the inflamed skin, and the injection of recombinant IL-10 speed up the resolution of pain in both male and female. So for that first part, we show that IL-10 signals to IL-10 receptor on sensory neuron. That IL-10 is necessary to promote the resolution of pain. But male produce more IL-10 than female. But so one of our next big question was, what's the cellular source of IL-10? And to answer that question, we perform high-dimensional flow cytometry. So high-dimensional flow cytometry means that all of our immune cells were stained with a panel of different markers. And then, based on the expression of those markers, we ask a computer to cluster the cell in an unsupervised way. So there's no human intervention. It's really we're just asking the computer to classify the cell, and that's what it came up with. And so cells that are very different from each other, like here you can see this green cells, they're very isolated and cluster very well. And the green, if we can see that we see that they're very high expression of CD3, high expression of CD45, and very low granularity, so that we know those are T cell. Another cluster of cell that is very isolated and so very different from the other one. It's this one. When we look at it, here's CD11B, super strong Ly6G is actually almost the only one expressing Ly6G, lot of granularity. So we know that those one are neutrophil. So in here, all of those cells are clustered together because they're much more closer to each other. And that's not very surprising because here we'll find monocyte macrophage with Ly6C low. We found dendritic cell as well. So we know that those cells are very close to each other in terms of origin and development. So using the same approach, we look at immune cell in the skin treated with saline, and in mouse skin in laboratories, there are very few immune cell in the skin. But when we inject CFA, we have a huge increase of immune cell, a huge infiltration of immune cell in the inflamed skin. So, and if you look very specifically at those cluster, you might notice that in female, that big red cluster is actually less innervated. And indeed, it's the only cluster when we see a sex difference with a higher number of cells in male compared to female. And those clusters are Ly6C positive, so very likely to be monocytes. Then to find which cell actually produce IL-10, because that was our initial question, we use the same approach with our IL-10 GFP reporter mice. And we found that several immune cells are actually able to produce IL-10 in the skin. That's relatively consistent with the literature. We found that they express in T cell, macrophage, monocyte, but overall, Ly6C positive monocytes were by far the primary source of interleukin 10. So here might be easier to visualize the number. So you can see that overall in both female and male, the number of GFP positive cells, so IL-10 producing cell, it's a small percentage compared to all of other immune cells. But when we look inside the percentage, inside those GFP positive cells, we found that the big difference between female and male are actually the monocyte, with male having much more monocytes that produce interleukin 10 than in female. We didn't see any difference for the other type of immune cells that could produce IL-10. And interestingly, when we compare pain sensitivity with the number of GFP positive monocytes, we found a correlation, which again indicates that those cells might be very important for the resolution of pain. And so overall, we found that male mice have more IL-10 producing Ly6C positive monocyte in the inflamed skin than female mice. So next, we wanted to remove those monocytes to see if they actually are the immune cell that promote the resolution of pain. So our first approach was to use clodronate liposome. So liposome, clodronate is kind of a bomb in a liposome, and all cells that have phagocytic capacity will phagocyte this liposome. And so then we'll phagocyte it, the clodronate will kind of explode like a bomb and kill the cell. So clodronate liposome will kill every cell that can phagocyte. So we treat the mice with clodronate, and then we've looked at monocytic marker, CCR2 and plaque8, and we see that strong reduction in both of those markers. We also see a reduction of IL-10 expression, and more interestingly, clodronate also impair the resolution of pain. But as I was telling you, clodronate is really not specific for monocyte. It will actually kill most of the immune cells that can phagocyte, so including macrophage, neutrophil. And so we wanted to have something more specific. So then we turn to a genetic model using Lyme's DTR. So a Lyme's Cree mice, which are relatively specific to monocyte, that we cross with a diphtheria toxin receptor flox mice. And so upon injection of diphtheria toxin, will kill all of the cells that express Lyme's, which are mostly monocytes, and mostly monocyte for now. And we see that when we treat mice with CFA, they develop pain, and after pain was developed, we treat them with diphtheria toxin. And killing this monocyte impair the resolution of pain, as that was true in female and in male, and that was associated with a reduction of IL-10 expression as well. But as I say, I'm not saying it, but yeah, Lyme's M3 is not super specific to monocyte. It is also expressed in subset of neutrophil. So we wanted to try another approach. And so here we use an anti-CCR2 and depleting antibody. And that is pretty specific to a monocyte that express a high level of CCR2 and do need CCR2 to exit the bone marrow and to go into the circulation. Same, we treat mice with CFA. They develop pain, and then pain starts to resolve, and we inject our antibody to deplete the monocyte, and in depleting the monocyte will again impair the resolution of pain. So we wanted to make sure that the monocytes were actually depleted. So we perform another high-dimensional flow cytometry. Here we change the marker. So here we select the marker to really differentiate between monocyte-derived macrophage, and tissue-resident macrophages. So for example, we identify this population of tissue-resident macrophages that have strong expression of CD206, MERTK, and CD163. While monocyte, that's mono 1, 2, 3, we found 3 clusters. They have much higher expression of Ly6C and CCR2, and very low expression of CD163. So that gives us good indication that this one were monocyte, sorry. And we also have monocyte-derived macrophages that are expression of, so intermediate expression in terms of CCR2 and Ly6C, and also have a strong expression of F4H. So looking at that, we saw that our approach with the anti-CCR2 really deplete monocyte, but did not affect monocyte-derived macrophages or tissue-resident macrophages or other type of myeloid cell. And if we look more specifically at the GFP positive cell, that's the one that expressed IL-10, we saw that the only population of IL-10 producing immune cells that we decreased was actually the monocyte. So that shows that we pretty specifically deplete monocyte without touching other immune cell, and that was sufficient to block the resolution of pain. So then to see if it was really IL-10 from the monocyte that is crucial for the resolution of pain, we cross the Lyme's M3 with IL-10 flocks. So in those mice, monocyte cannot produce interleukin 10, and that also completely block the resolution of pain. So then we wanted to know a bit more about the identity of these different monocytes. As I said earlier, we saw that it was potentially several clusters. So we did another high-dimensional analysis when we identify 4 different clusters. We named the cluster based on the kind of specific expression of some marker. So yeah, this one is labeled monocyte, but it's really a macrophage. It's a macrophage cluster. And so then we look at the which of this cluster is GFP positive or negative to see which one is producing IL-10. We observe that cluster monocyte, the monocytes that express also CD206 have the highest production of IL-10. But they are really monocytes with the high expression of CCR2, no expression of CD163, high expression of Ly6. And so the CD206 positive monocytes are really the main driver of IL-10 production among those monocytes. And interestingly, when we compare this cluster between sex, this is only the CD206 monocyte that shows sex difference with higher number of male compared to female. So far we found that IL-10 producing monocytes are necessary for the resolution of inflammatory pain, and that IL-10 producing CD206 positive monocytes are higher in male than in female. And so the question that we get from that is that it's nice to see to identify a mechanism that regulates the natural resolution of pain. But can we enhance that mechanism? Can we increase the number of monocytes to increase the resolution of pain and potentially treat chronic pain? So that was our first question after that. And the second question is that, is this mechanism clinically relevant? So we'll first look at the first question. Can we leverage that pathway that we identify to accelerate the resolution of pain? So we look into the literature, and we've been interested by specialized pro-resolution mediators, which are lipid mediators that are known to induce the resolution of inflammatory response. And after literature search, we decided to try resolving D1. We tried other one as well that didn't work as well, to be completely honest. But so with resolving D1, we saw that when we treat mice with resolving D1, we increase the number of GFP positive immune cell, which means IL-10 producing cell. And that increase was specific to the monocyte. And here, when we treat mice with resolving D1, the sex difference in monocyte that produce interleukin 10 disappear. We look at different type of cluster, and we found that that cluster, the CD206 positive monocyte, is actually the one that is the most increased by resolving G1. That's very interesting because that's the cluster that both have the highest production of IL-10 and is sexually dimorphic. If we treat mice with resolving G1, that accelerates the resolution pain. That was true in both male and female, and actually treating with resolving G1 erased the sex difference in the resolution of pain. So then we wanted to know if the effect of resolving G1 is really dependent on IL-10 producing monocyte, or if that is just a collateral effect of the treatment. So we treat mice with CFA, they develop pain, then we treat them with resolving G1. So, vehicle mice were here in black. They show the regular resolution of pain. When we treat them with resolving G1, we have this huge acceleration of the resolution of pain. But if we do that in mice where the monocytes cannot produce IL-10, it has absolutely no effect, showing that the pro-resolving effect of resolving G1 is dependent of IL-10 producing by monocyte. So until now, we've done our experiment with mice that were treated with CFA, so complete flood adjuvant, which is a solution of dead bacteria. It's a great model. It induces strong inflammation, but we can question its clinical relevance. So we wanted to make an injury model that is more likely to mimic human condition. So to do that, we induce in mice a physical injury with an incision done with a scalpel. And then we pair that with a session of stress. Mice get restrained for 2 hours. And then we test the behavior. And we did that because injury in human is often associated with stress. And so when we did that, injury plus stress induces strong pain hypersensitivity in mice. But again, we saw that male mice recover faster than female. And that faster recovery in male mice was associated with a higher upregulation of IL-10 expression in male compared to female. So as I said earlier, wanted to know if this mechanism is really clinically relevant. And so for that, we collaborate with Sarah Lindstedt at UNC, because Sarah follow a cohort of patients that experience a car accident, and then she collect the blood and sample after that, and follows the pain over 3 months. So interestingly, similar to our mouse model, we observe in men higher level of IL-10 compared to women after a car accident. So here we measure the pain. So is graphed differently than our mouse model. It's just a question, how is your pain from 0 to 10? So closer to 10 is higher pain. And we see that right after the accident, men that's in the emergency room, men and women have about the same level of pain. But when we look at 2 and 3 months after, the resolution of pain was much more efficient in men compared to women. And we did a correlation that showed that the level of IL-10 actually predict the resolution of pain. We also observe a higher level of monocyte in men compared to women after accident, and that the proportion of monocyte were also predictive of the resolution of pain. And in mice, we could not directly assess that the monocytes were the cells that are producing IL-10, but we found a strong correlation between the number of monocytes and the level of IL-10. And we did not find any correlation for every other immune cell type that we've looked at. So this data show that in human and mice, IL-10 producing monocytes correlate with the resolution of pain, and IL-10 producing monocytes are higher in males than in females. So we have really a strong similarity between mice and human. So because of that sex difference, we wonder if the sex hormone could play a role in this difference. So you may remember that in introduction, I told you that women are more likely to develop chronic pain than men. And it was actually a very interesting but small study looking at people that undergo hormonal gender transition. And when male become trans female, the pain get actually worse. But for females that become trans male, the pain actually improve, suggesting that that the sex hormone will play a major role in the resolution of pain. So we wanted to mimic that in our mouse model. So we use female with sham surgeries that have regular hormonal level, ovaectomy to remove the production of estrogen, and ovaectomy that we implement with testosterone pill, so which actually dihydrotestosterone, to make them hormonally male. And in male, we also have the sham control and male with orchidectomy with low level of testosterone. And then we did pain behavior and flow cytometry in the inflamed skin. So in female, first we show that ovaectomy, so the reduction of estrogen, did not do much for the pain, but addition of dihydrotestosterone improves the resolution of pain. When we look at the immune cell, we saw an increase in IL-10 producing immune cell with the addition of testosterone. And we saw also that and testosterone also increased the expression of IL-10. And more specifically, testosterone increased specifically the monocytes that produce IL-10. And you also and that increase was mostly driven by the increase of CD206 positive monocytes. In male mice, we observe that orchidectomy, so reducing the production of testosterone, drastically impairs the resolution of pain. And removing testosterone was also associated with a reduction of the number of monocytes that produce interleukin-10. And interestingly, it was not statistically significant, but really close to also show that reduction of CD206 positive monocyte, which, as you can see, that's the only population of monocyte that is really decreased after orchidectomy. So because those were systemic model and genetic model, so we try if we can have pharmacological validation. So we treat mice with flutamide, with an androgen receptor antagonist that is used for people that want to switch gender, and for men that want to become trans female. And blocking the androgen receptor slow down the resolution of pain and reduce the expression of IL-10 in the inflamed skin. And then to be much more specific, because so far our manipulation were at the systemic level, we wanted to know if the testosterone were acting directly on monocytes. And so for that, we generate a new transgenic mice when we specifically remove the androgen receptor from the monocyte. And removing androgen receptor completely block the resolution. Removing the androgen receptor specifically from the monocyte completely block the resolution of pain. And interestingly, removing the androgen receptor from monocyte did not impair the number of monocytes that infiltrate the skin, but it really just reduced the production of interleukin 10 from those monocytes. So in this last part, we show that androgen hormone like testosterone facilitate the resolution of inflammatory pain, and that androgen hormone like testosterone increase the infiltration of IL-10 producing monocytes in the skin. And so to conclude, what we show is that in model of inflammatory pain, we have monocyte, especially CD206 positive monocyte that produce IL-10 that signal directly to sensory neuron by its receptor to reduce neuronal stability and promote the resolution of pain. And that mechanism is less efficient in female mice than in male because testosterone increase the production of IL-10 from the monocyte. And we can switch that by manipulating the level of testosterone. And strikingly, we found that in human, after car accident, the same correlation with slower resolution of pain in female with lower number of monocytes that produce IL-10. So I want to thank all of our sponsors that make that research possible. But really want to thank you for your attention and your invitation. I want to thank everyone in my lab that contribute to that work, especially J1 Sims, our main PhD project, my collaborator in Houston and in North Carolina and an MSU. And I wanted to also to say that we'll be looking for postdoc to continue to work on neuronal immune cell communication. Thank you so much for your attention.